Enzymes are proteins that act as catalysts to speed up the rate of reactions and processes. They have three characteristics. Number one, their function is to increase the rate of a reaction. Number two, most enzymes act specifically with a single reactant called a substrate to form products. And number three, enzymes are regulated from a state of low to high or high to low activity. There are three different kinds of enzymes: digestive, metabolic, and food. Digestive and metabolic enzymes are produced by the cells of the body. Food enzymes are from the food that we consume. They have a part in digestion, in the breaking down of vitamins or nutrients ingested, in building cells, and in reactions that transform energy so that it is usable. Like all proteins, enzymes are long linear chains of amino acids that fold to produce a 3D product. Each amino acid sequence produces a specific structure with unique properties. Enzymes are very specific and only work with certain substrates. Complementary shape, charge, and hydrophobic characteristics of the enzymes and substrates are responsible for these specific decisions. The two major parts of an enzyme are the active site and the allosteric site. Both are rich in extremely complex arrangement of proteins. The active site is the part of the enzyme that binds to a substrate through hydrogen bonding. When a substrate is bound to an enzyme substrate complex is formed and the enzyme is ready to begin its job. The allosteric site majorly affects what happens at the active site. This site is able to bind to other enzymes called coenzymes or non-organic matter called cofactors, like iron or zinc. The binding of these substances in the allosteric site regulates the availability of the active site to do work. The active role of a cofactor is to either activate the protein by changing its geometric shape or participate in the overall reaction. Differing parts of enzymes allow them to perform different functions. There are several differing minor parts of the enzyme as well, but they are not as important as the active or allosteric sites. The polypeptide or the protein part of the enzyme is called the apoenzyme and can be inactive in its original structure. The inactive form of the apoenzyme is known as the proenzyme. The proenzyme contains extra amino acids in the protein which are removed. This allows the final tertiary structure to be formed before it is activated as an apoenzyme. The relationship between the cofactors and the apoenzymes vary. Sometimes the bonds are loose and only occur during a reaction. Other times they are bonded together by strong covalent bonds. Lipase is an enzyme that acts on lipids to create glycerol and fatty acids. Most lipases act on a specific position on the glycerol backbone of a lipid substrate. For example, the human pancreatic lipase is the main enzyme that breaks down dietary fats in the digestive system. It converts triglyceride substrates to a monoglyceride and two fatty acids.